Hello and welcome on Windows 8 Studies. Today we'll be learning about epigenetics. In epigenetics, the word epi is Greek derived, which means on or above the genome. So this is the literal meaning of epigenetics. So now let's dive into the details of epigenetics, how this field emerged, and why was there need of epigenetics? What is this field explains? What are the questions which are solved by this field? So for that, we have to look at the Human Genome Project. The first draft of Human Genome Project was published in 2001. At that point of time, the president of US stated that today we are learning the language in which God created life. So this statement shows how the researchers at that point of time believed that DNA sequence is our final key to decipher all of the questions that couldn't be solved in developmental biology and genetics. The questions like why the twins which have same DNA sequences differ. They differ phenotypically. For example, one twin can have blue eyes while the other twin can have black eyes. Why they differ behaviorally? One could be rebellious while one could be obedient. So the twins have same DNA sequence, the same set of the genome. Still, they differ while the science tells us that they should be 100% same, right? So this question could not be solved by genetics. So this question was put in epigenetics. At that point of time, all of the questions that couldn't be solved in developmental biology, cell biology, genetics, something which we don't know, all of that was put in epigenetics. And at that point of time, epigenetics actually meant that something which is beyond genetics, something which can't be explained by genetics. This term was coined by Con, Conrad Waddington in 1964, I guess. So in 1964, Conrad Waddington coined the term epigenetics, which some point of time at that point of time was actually meant that something which is beyond genetics, which can't be explained by genetics, is what is included in epigenetics. But now it's actually on or above the genome. Something is occurring on the genome due to the environmental changes, due to the other factors, and that is included in epigenetics. Actually, the definition of epigenetics has been varying over the decade, over the past 50 years. For now, let's just assume that non-sequence dependent inheritance is epigenetics. I'll be elaborating more about epigenetics. Till now, just get a note that non-sequence dependent inheritance means epigenetics. So this questions were the question like individual, will it have character, will certain character, or say eye color, or say hair color, or say hair type, all those things, would it be inherited from paternal parent or from maternal parent? This, what is there, what is there which decides which from which parent I'm gonna inherit that factor? So there should be something, right? So all those questions could be answered by epigenetics, all those questions. So let's now take a look at what happened in past, which made scientists aware of such a beautiful thing. So there was a very unfortunate event in World War II, which actually led to case studies by epidemiologists at that times, and that event is known as Dutch hunger event. So there was a drought, an extreme cold period in Western Netherlands, and at that point of time in the Western Netherlands, the World War II was just ended, but Germany still had control over over the lands there, and they blocked the food the food supply of the Western Netherlands. So German blockade resulted into very bad famine. It was so bad that people went malnutritioned. People burnt all of the furniture that was available in their house. They ate tulip birds. They That was a drastic drought period. So in the Dutch hunger winter, epidemiologists of that time studied newborns. How they studied newborns? They actually weighed them. So they weighed newborns of two types of mothers. The mothers who were poorly fed in the first trimester. Mothers who were poorly fed on the first trimester, and later they were properly fed. Maybe those mothers were like the mothers who were conceived at the just around the end of World War, though. So they were at the first trimester, at the first 3 months. They were on the shortage of food, but after that they were properly fed, while the other mothers which were poorly fed in last trimester. So the babies born by the mothers who were poorly fed at first trimester, but then normally fed, those babies were normal in weight. While the babies which were poor, while the mothers who were poorly fed, those mothers gave birth to the babies which were small and had petite figure. Okay, so now this kind of makes sense, because at that point of time researchers believed that all of the growth happens at the last trimester. So if the mother was poorly fed in the last trimester, baby is gonna be small. But if they are fed properly in the last trimester, the baby will be normal. But those epidemiologists, they had the chance to study those individuals over the decades. So they studied those babies further for the decades, and they found out that the small babies, they still remained malnutritioned throughout their life. So this doesn't make sense, right? Because even when they are getting standard diet, the drought has been ended. There is no control of Germany over that area, and there is no food shortage. They have been given proper diet. They are standardly fed. So then why are they still malnutritioned? Because of the something that happened even before they were born. They are still malnutritioned. Well, the babies, which at the point of birth seemed normal, they were actually vulnerable to obesity. They showed high obesity rates. So the seemingly normal babies were also vulnerable. They were also not normal. So there is something which is causing of the all of those something unexplainable things. So there something wrong happened. Something wrong happened which made the seemingly normal baby which should have been normal, but something wrong happened. Something wrong was due to the poor diet of mother. Something wrong happened which made those babies deviate from the normal babies. Okay, but not just matter of concern is not just that something wrong is happening, but matter of concern is that when is that something wrong is happening, when is the defect or something that went wrong is happening, when. So what matters is what happened. What happened when the mothers were poorly fed, which made babies go malnutritioned or highly obese, while when that thing happened also matter, because if that happened in the first trimester, babies were normal, seemingly normal, but they were vulnerable to obesity, while if that happened in last trimester, they were actually malnutrition throughout their life. So alongside the thing that happened, it all matter of concern is the timeline when that thing happened. So timeline is also important factor. So this is all explained by this is all a matter of concern in epigenetics. Something beyond is same thing beyond genetics is happening, something which is affecting the DNA, and those two kind of outcomes we can see. So now, what the Dutch hunger winter made the researchers realize was that at like over several decades, researchers thought that DNA is more like a template or say a mold. Okay, where you would imagine if the mold is like this, you will put wax in that mold, and the artifact you're gonna get would be something like this. So the way mold is, the way artifact would be. Okay, as simple as that. But it's not as simple as seems. Then it got to know that due to this Dutch hunger winter event, that DNA is not just like a template, DNA is more like a script. Okay, now imagine that there is an audition. Okay, in that audition, all of the auditioning actors will be given same script, right? But only one will be selected. Why? Because of the outstanding performance. Why that one was selected? Because of the outstanding performance given by that actor. But this doesn't mean that other actors didn't act. They acted, but differently. So the same script given to different actors generating generating different outcomes. Okay, assume that it was given to three actors, and it has the three actors have their own acting, different actings. Okay, so outcome matters on the readers. Okay, so the possibility or capability of same script giving the different outcomes is considered here. Okay, so let me give you another example. For example, you are in the class, and your professor gave you an assignment to the class. In the class there are 5 students, and you have been given an assignment, and you have to turn in that assignment after 3 days. Okay, so you all turned in that assignment after 3 days. So we have 5 assignments now. 1, 2, 3, 4, 5. 5 assignments from 5 different students. Now the teacher says that read your assignment as well as others assignment and submit me a report of your comprehension. Okay, submit the report of the comprehension, what you have comprehended from the 2, 3, 4, 5 different scripts. Okay, so now one I read my own script. Let's assume I'm one. I read my own script, I read my fellow batchmate scripts, and I got different type of things. So when I read the script written by 2, I'll comprehend it in the X manner. When but when the same script is read by student number 3, it will comprehend in the Y manner. Okay, so the comprehension will differ from student to student. Now the script is same, the script is written by 2, that is same, but it is, if it is comprehended by on the 2, the person 2, it won't be, it will be say X type kind. Okay, but when number 3 comprehended it, it will be Y kind, and when I comprehended it, it will be M kind. Okay, so same script is giving different outcomes on who is the reader. You got it? So in the epigenetics we have different writers, we have different readers, and again one term will come, different erasers. Okay, I'll be explaining all those terms in upcoming videos. For now, just assume that the script has been if the script, there is single script, has been read by different readers, it will give different outcomes. So the script is DNA here. Okay, so let me show you. DNA is the script, bases are the word, the nucleotide bases are the words. Okay, so the script is made of words, and the script is read by readers. Okay, and the readers, or you can say that actors are the reading it. Okay, actors means genes. Okay, the genes would be the actors, or oh sorry, genes, no, 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 not the genes, cells. Cells will be reading the DNA, right? Cells will be reading the DNA, or cells will be performing after reading the DNA. So that's why cells are the readers or actors here. The terms I told, writer, reader, and erasers, in terms of enzymes, those are different. I'll be explaining that. So please, this is not the matter of concern here right now. I'm just telling you that your life is a movie, and DNA is the script given to you, to given to your cells. Cells are the actors, they will be act, and depending on the which cell type is reading the DNA, the outcome will be different. But outcome gonna be different on not just depending on the cell type, but also the director, right? If director tells you, your director tells you that do this and do that, it tells this to different actors, like different cell types will behave on the command of the director differently, right? So the director here is epigenetics. This is a small analogy just to get I'm just explaining all this in layman terms just to get a better idea of what is happening in the cell. So different cell types are going to decipher the same DNA sequence differently, because not just because the cell type is different, but because the director, the epigenetics, will be coming in there. Okay, so genetics is just like screenwriting. Whatever you have been provided, you are just writing on the screen for the movie, and while the director is epigenetics. So this was all about epigenetics, the basics of epigenetics. What is epigenetics? You could say epigenetics is also considered as non-sequence dependent inheritance. So the sequence dependent inheritance is considered in genetics, but non-sequence inheritance is considered in epigenetics. In epigenetics, we consider, like, it's beyond the sequence of DNA. It's not just DNA sequence matters, but what is on that DNA sequence, the tags on that DNA sequence, that all matters. The tags could be methyl tags, the tags could be acyl tags, or the phosphorylation, or sumoylation, or say acetylation. So non-sequence dependent inheritance is epigenetics. So this was all about epigenetics, the proper definition of epigenetics, which is like modern definition, which is very close to the today's world. I'll be explaining all that, as well as DNA methylation, nucleosomes, everything in the upcoming lectures. Till then, see you. Bye bye.